Okay, great. Well, thank you all so much for making it this Friday afternoon for a special Compass seminar. I'm really delighted to have Carly Buchwald visiting us. She's a colleague of mine. We went to grad school together in the MIT Woods-Hole Joint program, both in the chemical oceanography program, and she's coming to visit us from Dalhousie University in Halifax, Nova Scotia. And she works on nitrogen, and she's a microbial biogeochemist, looks at nitrogen cycling in a lot of different aspects, also works on kelp aquaculture and alkalinity enhancement. So she's going to be touching briefly on those and digging deep into the nitrogen cycle. So, and she's visiting us here on a sabbatical, so if you folks have questions after her talk, she'll be here through next week. So just let me know if you'd like to set up a time to chat with her more about her research. So thank you for visiting and thank you for coming.
Yeah, thanks so much, Kim. So this is my first sabbatical, and I wanted to go away for longer periods of time, but I have small children and a husband that works in person, so I'm kind of just doing a little bit of tour to kind of get myself reinvigorated in science and talk. I'm really interested to talk about the research going on in different places. I had a great meeting over in the experimental hatchery this morning on aquaculture, which is something I do now. So I'm here just to talk science. So I told Kim not to do the full intro because I always start, this was working. I think I have to click on the screen. Yeah, I always start a little bit about my research path. It's not a very complicated one. I was born in Massachusetts and I grew up in Massachusetts. I start with that because often people think I'm from Canada. I'm not from Canada. I went to MIT as an undergrad. And while at MIT, I got pretty into oceanography and I kept wanting to go down to Woods Hole. And so I kind of actually the whole time I was there wanted to become a joint program student. So I did end up doing that. I looked at some other oceanographic places, but I did end up just basically moved down to Woods Hole. But then my advisor, while I was getting my degree, moved to Stanford. So that was the first chance I did get to experience a different area of the country. And while I was at Stanford, I was talking about doing postdocs at different places, but the person that I ended up talking to most was actually at Harvard University at the time. But he was in the process of being, he just got actually the job at Hui that my advisor left behind. So then I basically ended up doing a postdoc at Hui. So I ended up spending a lot of my career at Woods Hole Oceanographic Institution. And then from there, I got my job at Dalhousie, and I started there in 2016. So I've been there 9 years now, which almost. I had 2 maternity leaves in that, but it doesn't really feel like I've been there that long. And I am finally Canadian as well. So, okay, so I'm going to just give a brief overview of why I do the research I do, and then I'm going to talk about a few of the different areas I'm studying. So I think most people already know, but I still start off with it. Why do we care about nitrogen? So I care about nitrogen for a few reasons. One, because it's the major nutrient needed for all life in the ocean, right? So it supports the ability for phytoplankton to grow and take up CO2 into organic carbon. So I really care about nitrogen because it fuels this carbon export. Nitrogen is also really interesting because it's one of the elements in the ocean that has multiple species. So you have your fixed or reactive species, nitrate, nitrite, ammonium, and dissolved organic nitrogen. And those are the nutrients, so those are the things that ocean grows. But what's interesting about those is that there's special microbes that actually do the transformation between these species and get chemical energy from that. So I actually study nitrification, one of the processes that does that. Nitrogen is also in its gaseous form. So nitrogen gas is 78% of our atmosphere. Nitrous oxide, while in low concentrations, is still a very potent greenhouse gas in the environment, and it is a byproduct from some of the microbial processes I study. Nitric oxide is also a pretty important intermediate for some of those processes. And so I always show this. This is pretty old at this point, but basically if you focus your attention to the pink area, that's basically the area of the ocean that's limited by nitrogen. So ultimately, nitrogen is one of the major limiting nutrients in all of the ocean. So that's why we can make statements saying that it controls export, because it really does control export in a lot of the ocean. This is a plug for my kelp aquaculture work. I got into studying seaweed because it requires nitrogen. So now we have a facility where we grow baby kelp. And it's kind of hard to see here, but this is a PVC pipe with string along it. And if you look at these pieces of string that are hanging off, These are all little baby kelp. And then we take that string, we wrap it around a string in the ocean, and then it grows up into like big, beautiful kelp. And that's in March. We don't usually harvest until May-ish, end of May, and so that kelp will get a lot bigger. But I got into kelp aquaculture because kelp also needs nitrogen. So this community of aquaculture cares about nitrogen in the sense that they need it. Other areas of aquaculture, such as fish, care because they produce too much of it. So that's just an aside. And then not only is nitrogen important in the ocean, it is also one of the elements that we are drastically changing its cycle in the environment. So humans are adding fixed nitrogen. We grow nitrogen fixing crops to support food production. We do a lot of fossil fuel burning, which actually releases NOx's into the atmosphere. And then most importantly, we do the Haber-Bosch process, which is basically taking nitrogen gas and fixing it into ammonium fertilizer. If you look at human population growth in blue and the total amount of reactive nitrogen or fixed nitrogen in the environment, the ability for the human population to grow as much as it has is actually directly because our ability to grow more food. And the way that we grow more food is by producing more nutrients. So it's important to talk about because in the ocean, so this is actually showing where this red is this human fixed nitrogen. So this is one side is on the land and the other side is in the ocean. And you can see a lot of this human fixed nitrogen is going into the land. But then if you look at where nitrogen is coming into the ocean, so either from atmospheric deposition or rivers, you can see now half of that nitrogen that's coming in is produced through human impact, so through human industrial fixation. So we're actually like, in an oceanic sense, we're doubling the amount of fixed nitrogen that enters the ocean than we had previously. So it doesn't look insanely impressive in this picture, but that's just something to note that we really are changing our coastal environments by adding a lot of nitrogen. And that matters because of eutrophication. So this is something that you guys are probably used to here in Florida, but anywhere that you have nitrogen coming into a coastal ecosystem, if you get a lot of nitrogen at once, you can have algae blooms that will grow on that nitrogen very fast. And when they die, which blocks out all the light, and when they die, the microbes that respire that organic carbon consume all the oxygen in the water column. And this creates, scenarios that look like this, which leads to fish kills, harmful algal blooms, just algae kind of all over your coastal beaches where people might want to hang out. So, ultimately, there's kind of like two things. So to me, nitrogen is kind of this balancing act. We need it for the ocean to take up carbon and make food in the ocean and control climate. But also in coastal areas, if you have too much of it, it causes issues. So to me, like, nitrogen is interesting because it's this balancing act. Like, sometimes we're like, yes, nitrogen is limiting, and sometimes we'll say nitrogen's too much. So basically, that kind of boils down my lab goals in kind of two main areas. One is understanding, fixed nitrogen's role in the biological carbon pump and how it controls climate, and then also tracking this fixed nitrogen in coastal environments. So now in Nova Scotia, I look at places like finfish aquaculture, wastewater treatment plants, and fertilizer runoff. So where the nitrogen's going in coastal environments. So how do I do this? So how do I track nitrogen? One is just measure it, but what I'm talking about today is more how we use stable isotopes. So I always also do a quick marine nitrogen cycle. So I kind of teased at parts of it. But the marine nitrogen cycle, in blue, we're going to talk about where fixed nitrogen is coming in. The red are the outputs where fixed nitrogen is leaving, and green is this internal cycling that happens. So one of the major natural sources of fixed nitrogen in the ocean is through nitrogen fixation. So I'm not talking about runoff from humans, but then this fixed nitrogen is fixed by these phytoplankton that can fix it in their bodies. they can then, when they die, their bodies are regenerated, and you regenerate ammonium, and then you have nitrification that oxidizes the ammonium to nitrate. These species of nitrogen are forms of fixed nitrogen that can then be taken up by other phytoplankton that can't do nitrogen fixation, or even the ones that can but don't want to right then. When those organisms die, they sink. So most of the remineralization actually happens while these organisms are sinking and dying in the ocean, and that's where you get a lot of nitrification or remineralization in the ocean. But some of the outputs are through burial. I also didn't mention yet, but it's going to come up a few times in this talk, is that nitrous oxide is a byproduct of nitrification. And so that's another reason that we care about nitrification in the ocean. Also, some of these species might end up in sediments or in anoxic regions in the ocean, and that's where you get denitrification and anemox, so anaerobic respiration that respires nitrate and removes it to N2 gas. I want to understand kind of the rates of these processes in the ocean. So how do we do that? So how do we measure the magnitude of these arrows? If I want to know how much nitrification is happening somewhere, how do I do that? So classic ways to do that have been bottle incubations. So this is incubators on the back of a deck of a ship. I think this is Eastern Tropical South Pacific. And you can see, this is running seawater in here. You can see the bottles in here. And a bottle incubation is basically where you take some water from the ocean, you add a tracer. So the tracer will be what process you want to track. For example, if you're tracking ammonium oxidation, you'd add ammonium and you'd watch the ammonium get converted to nitrite. And then you measure that change over time, and from the slope, you get a rate. And so this is kind of the classic way of getting rates in the ocean. And you can do this for a host of different processes in the ocean. As some of you who may have done rate incubations before, there's actually a host of issues with it. One, they're really time consuming. They take up a lot of space. I think that was one of the biggest incubator farms I've had. They take up a lot of space. And the ocean is so spatially and temporally heterogeneous that to get the amount of depths and stations you want, it just becomes hundreds and hundreds of bottles. And so it's really hard to get the resolution that you want. Also, these bottles sometimes do not really mimic the environment perfectly. It's hard to get the right light levels and temperature levels, and some microbes don't like being in the bottles, some do. And so there is, some idea that these are kind of relative rates and not really exactly what's happening in situ. So that then leaves me to stable isotope, which is basically a tracer. I think everybody here probably knows what an isotope is. It's something with the same element in different mass. I'm going to be talking about stable isotopes. So they exist naturally. They don't decay away. And so for nitrogen, there's two major stable isotopes, N14 and N15. In nitrate, you also have an oxygen atom, which has three major stable isotopes, 16, 17, and 18. We usually focus on the ratio between the 18 and 16, the two major of the stable isotopes. And when you measure an isotope profile in the ocean, this was one of the first ones measured for both nitrate, oxygen, and nitrogen at the same time. And you can see in the deep ocean, where nitrate is a bigger, steady pool, you get a really constant isotopic value because you're not getting much change in that nitrate pool. When the nitrate is in the surface ocean starts getting taken up by phytoplankton, you see this enrichment. So you see a signature of the fact that there was biology going on there. And then this signature is how we could figure out what's happening and at what rates. Those signatures are created by isotope fractionation. So isotope fractionation is just when the different isotopes react at different speeds. The fractionation factor will just be the ratio of the reaction rate of the light isotope over the heavy isotope. An isotope effect is just another way of writing it, which is just that fractionation factor minus 1 times 1,000, so you get a number versus a fraction. So the way this works is if you have a microbe that's doing denitrification, it sees a pool of nitrate of 50%, 14, and 15, which it'll never see something that enriched, but just for example's sake, it will preferentially reduce the N14s over the N15s. So then it's producing nitrite that has a more N14s to N15s, so it gives you a product that's depleted and a substrate that's enriched. And these fractionation factors is how we can tease apart what isotopic processes are happening in the ocean. So basically, for every one of these lines, they're carried out by different microbes, and they each have specific fractionation factors that we can measure. So one of the things that we do in our lab is actually, and this actually comprised a lot of my PhD, is growing these types of microbes and figuring out their fractionation factors, and actually figuring out what are controls on their fractionation factors. This student right here, Jennifer Butler, she was using a chemostat, and we were tightly controlling growth rate to see how actually growth rate affects the fractionation factors that you get. Okay, so why use stable isotopes? So I kind of already got you there, but basically we do this to get rates from static, cost-efficient, easily taken measurements. So it's basically we build 1D models, and so my first case study that I'm going to talk to you today about is about a model that we built in the Labrador Sea for measuring rates of new and regenerated production. The thing about these models is they do require a very in-depth understanding of these microbial processes and the associated isotope effects. And so that's why my lab also grows these microbes, or collaborates with people like Kim or Hillary, like people that are trying to understand these microbial systematics of what the microbes are actually doing in the ocean, because that all builds these models are pretty complicated, and we use all the knowledge we have to build them to produce the isotope profiles that we see. So this is just a quick example of how this works, and actually I did this is actually deep sea sediments. So this is from a IODP core in the North Atlantic, and this was just pretty because you just get really pretty profiles from something like this. But so the dotted line is oxygen. The blue is my nitrate concentration. The green is my nitrite concentration, and the blue is ammonium. And so you can see in concentration, you get a really pretty profile. And just from concentration alone, we can make some assumptions about the processes that are happening. Now, the one unique thing in these anoxic regions is that you can have areas where you have nitrite oxidation and nitrate reduction happening very close to each other and co-occurring. And that's something where when you have two processes kind of doing opposite effects, in a concentration, you can't always see it. But in an isotope, you can tease that apart. So then this is the profile of the N15 nitrite, O18 nitrite, N15 nitrate, O18 nitrite. So now I'm building together, and then this black is actually, we sometimes use a parameter that is the difference between your nitrite N15 and your nitrate N15. And for this example, I'm not going to go through the details, but I was just going through it for the method. Then we can basically build a model that comes out with a rate profile like this. So purple is my ammonium oxidation rate. For example, I only allowed ammonium oxidation to happen in the very tops of the sediment where there was oxygen. So I am using some of my knowledge of the chemistry and what could happen to build these profiles. But then you can see in these dotted lines, you have this co-occurring nitrate reduction and nitrite oxidation happening at the same time. And you kind of need that to explain the isotopic signatures that you're seeing here. But if you didn't know that, you wouldn't actually need that. These red and green lines here, you could actually assume that you just had nitrate reduction and then nitrite reduction after that, and you only have a tiny bit of nitrite oxidation above. So basically, the isotopes are what's kind of telling us something a little bit different than just the concentrations alone. But it's a similar principle too, that we build these models and get so now I have this beautiful rate profile versus, for example, in my bottles, I would maybe have 3 depths that look something like that, and I'm building something that looks something like this. And that's kind of the crux of why I do what I do, basically. And then the second way, when we think about kind of the second goal of my lab is tracking fixed nitrogen in the environment, isotopes also fingerprint things like anthropogenic sources or different sources of nitrogen. So this was a project one of my master's students did in, so this is Dalhousie right here. We're on a peninsula. There's something called the Northwest Arm right next to us. And she sampled at 3 locations in the arm just with the bucket sampling. And she actually started this as her honors project. And we were just seeing if we knew that there's some sewage overflows. So that these sewage overflows only get sewage when the wastewater treatment plants shut down. But we were like, can we see any of this signatures in the arm? And so you can put together a plot like this. So for nitrate is interesting because it has its N15 and O18 signature. If the symbols here are from her sampling locations, the boxes are just known values. So for example, nitrate that comes from rain has a very high O18. And so we know if we're seeing something that's from precipitation, it would look somewhere up here. Nitrate fertilizer also tends to have a higher O18. Wastewater and organic nitrogen tends to have heavier N15 than these things that have come from ammonium fertilizers and then are oxidized. And then this box is kind of our oceanic, is what most nitrate in the ocean falls within. So you can see while we were there, we actually mostly just saw the oceanic signature. This is, connected to the ocean. But there were times of years that we saw some of these elevated values. And while it actually looks like it's falling on the nitrate fertilizer, we actually think it was actually mixing with precipitation because these were actually happening when there was ice coverage. And the ice would, I think, accumulate the atmospheric nitrate, and then when it would melt, we'd see these high O18 values. But you can actually use more sophisticated models to actually tease apart what's going on here, but this is just an idea of how you can use the isotopes to kind of track things. And then we really weren't seeing much signature of wastewater. We actually think that's one is coming in a lot as ammonium. It's probably getting taken up to the phytoplankton really fast, and we're not really actually seeing it in the nitrate signatures. Okay, so that's really just an overview of what we do and why we do it. Now I am going to talk about 2 different case studies. The second one, I'm actually really just going to introduce what we're doing, but we actually have no results yet. But it's just interesting because it's topically important at the moment. Okay, so my first study is from the Labrador Sea. And so it's actually this black box down here. And this is work done by my PhD student, Britton Dempsey. And the Labrador Sea has a seasonal spring bloom. This is satellite images. So we were there, and this is from a cruise in 2022. These are, the dark red shows the bloom forming starting in April 29th to June 1st. You can see we were there at the end of May. So you can see by the time we were there, you can kind of see our stations on here. The bloom was dissipating at that point. If you do a integrative average surface chlorophyll A, so this is like integrated over this whole box, these are what the bloom looks like year to year. We were there in 2022, so it was actually one of the second biggest blooms we'd seen in the recent times. And then this green line is when we were there. So you can see we were there at this dissipation of the bloom. Also, when we were there, the bloom actually looked like pretty much a monoculture of Phaeocystis pouchetiae, which is a type of haptophyte. They're known for making a very gelatinous material. They clog up IFCBs and things like that. And it was it was very soupy, I guess is the word. We also had people out there measuring carbon export, looking, doing thorium measurements and deploying traps. People looking at marine snow production. And so we actually, a lot of this is still in the work. We're having a big data work. A lot of this is still preliminary. And we're having a big data workshop coming this June. But, preliminarily, they didn't really catch any big export events while we were there. But we were coming from the side of nitrogen, right? So I've already kind of mentioned this, but we wanted to get at export by understanding the ratio of the amount of nitrate that was taken up through new production and looking at how much recycled production was happening. And specifically, we wanted to know if there was any source of recycled nitrogen within the surface producing nitrate that would affect your new production estimates. And we really, and the other thing is we wanted to know how much resupply, so how much nitrate was coming in during the bloom that was supplying this new production. And the reason that we care about this is because part of the reason we're studying this region in the Labrador Sea, it's an area that's undergoing lots of change from climate change. And we kind of want to know, are we going to see any changes to our F ratio year to year, where we end up having more systems that have more recycled production versus new production and lower our export in those regions. So we're interested in the dynamics that could change some of these things. Also, resupply is affected by stratification. So if the waters become more stratified up there, it also could affect the amount of resupply that happens during these blooms. So if you were to try and estimate new production just based on the inventory of nitrite is there, you'd do something like this. So you'd say, okay, this is what the nitrite looked like before your bloom. This is what the nitrite concentration looked like after your bloom. And then you can get an estimate of nutrient production just based on the inventory of nitrate that went away. And that's what people have done in the past. And it can work to some extent, except for we know that we have things such as regeneration and resupply, so nitrite coming up during these blooms, mixing events, storms during these blooms, as well as that surface nitrification, so some of the organisms dying and releasing ammonium and being oxidized kind of within the surface. That's gonna change the amount of actual new production you would have estimated just based on your concentrations alone. So basically our remedy is to bring in the nitrate stable isotopes to complement a nitrate drawdown model and see if we can estimate the amount of resupply and regeneration. So this is kind of how the approach works. So this is nitrate over time. So this time is the, and then chlorophyll over time, and then your N15 isotope. So if you assume, before the bloom, you have pretty well mixed water column and high amounts of nitrate in the surface ocean. Once your phytoplankton start to bloom and grow, you get a depletion of your nitrate. While that nitrate is being depleted, because of fractionation, this surface nitrate pool is getting enriched. And we know something about the fractionation factor that causes that enrichment. Now, if you get some mixing events where the mixed layer drops and you get this, amount of resupply, we keep calling it, some nitrate coming back in, you can actually, what that's going to do is going to allow you to grow more organisms in the surface ocean there, as well as actually deplete your N15 signature. So now you're going to get an isotopic signature that looks less than what you'd expect it to be because of this resupply. And this difference is what we can use this difference in isotopes from what we expect it to be to what we see is what we can use to estimate what that resupply is. And so we initialize our model using World Ocean Atlas mean, so the nitrate concentration being well mixed before. Then we are here, so we allow, and these are actually the samples that we measure, so you can see the drawdown we saw. You can see that we actually didn't see complete drawdown. That's a whole other area of research of what was actually terminating this bloom. Were they limited by things? Was it grazers causing it? But either way, we know how much the nitrate was drawn down in our model. We run this model for the amount of days to get a seasonal integration over the whole bloom, because these nitrate isotopes that are from these nitrate measurements will integrate over that time. If we don't allow any resupply, and we were just to assume, nitrates getting taken up by phytoplankton by known isotope effects, this is what it would look like. So the red is the nitrate concentration and the blue is the isotope. So as nitrate's going away in the surface ocean, you get nitrate being produced that's enriched. And just look at where this blue is getting up to, right? It's getting pretty enriched in the surface as nitrate is going away. If we think about it in terms of Rayleigh space, or for people that study isotope fractionation, that's just the natural log of the nitrate remaining. But basically, it follows a line where the slope of that line is the isotope effect that you measure. So for uptake, the global average is about 5 per mil. And there's been many experiments with phytoplankton in culture too to say that it's something around 5 per mil. If we now look at the N15 compared to the O18, You also, we know about phytoplankton that they actually fractionate the nitrate equally for the oxygen and the nitrogen. So you'd expect them to fall along a 1-to-1 line. And that's what this is showing here. Now, in our scenario now where we allow resupply and regeneration, we have a couple things that now happen. Now you can see in blue, we don't see as much enrichment because we're bringing in, keep bringing in light nitrogen while it's getting enriched. You actually deplete, you dilute the amount of enriched nitrogen you see, so then you only see it getting a little bit enriched, and I kind of explained that in those first couple slides. You also see a depression of your apparent isotope effect. So now that the isotope effect that you measure actually looks like something lower than what you expected it to be. And then because of nitrification and regeneration, you also fall off of this 1-to-1 line because nitrification brings in new oxygens with a different isotope effect. And so now your relationship between N15 and O18 are not 1-to-1. So these are the two things in our isotope plots that tell us that we don't have a closed system scenario. So we optimize the model against these observations, and we end up modeling 5 different parameters, but we basically run the model until it fits the data the best. And so these dots are the data. And we end up, and so you can see that, our apparent isotope effect is now closer to something like 2.6, and that we do get this deviation where you're a little bit above the 1-to-1 line. Okay, so this is still pretty preliminary, and my student is running lots of sensitivities on these 5 parameters in now on this model. But you can see even preliminarily, like the red line is the this thing is, yeah, this works so much better. The red line is the model results. The red dots are our samples. And so you can see that if we include this is all in millimoles of nitrogen per meter squared per day, about 9 millimoles of nitrogen, 9 for resupply, 6 for regeneration, and then the total uptake of 32. And it also, the model it optimizes the regeneration isotope effect as well as that assimilation isotope effect. You end up getting these numbers. You can multiply that by, well, it's actually this minus this times 6.6, and you get an average carbon uptake. We call it a max because this is over kind of the whole 50 days, but this is the amount that could have happened. This number is larger than what other people on the cruise had measured so far, but we do think that could be related to kind of time scale differences in these measurements. And ours is really integrated over the whole bloom. But and like the other kind of main takeaways is that we for sure are seeing this resupply. So you do need to consider that nutrients are there is this diffusion of nutrients kind of throughout the bloom that are sustaining this bloom. And also there is some of this regeneration. So 18% of that new production is supported by regenerated production. And we also, like for the isotope people, is like these isotope effects were kind of inversely determined from these models, and they fall out within the ranges of what we see of phytoplankton in culture. Okay, so that is that. I'm gonna take a sip of water. Does anybody have any? Do you guys care if I ask questions in between? Because if anybody wants to ask anything on this, I could also wait to the end. Yeah, Sabrina, go.
First of all, super cool.
Yeah.
Second of all, so you multiply by 6.6. You mentioned that this bloom was primarily driven by these like gelatinous guys.
Yeah.
Do you think that they might have more carbon because they are gelatinous compared to some?
And actually, you know what? They totally have the C to N, and I probably could have actually got what the actual C to N of those organisms are. The thing that I should say though, and I think the jury is still even out on this, is that that's when we were there. This is seasonally integrated, so this is over that whole bloom. There is a chance that there were diatoms in the beginning, and then by the succession that we got there, now we're looking at a different group of phytoplankton. And they are working, so I didn't really say the satellite work is all done by a group at the Bedford Institute of Oceanography, which is part of DFO, or our Department of Fisheries Oceans, very similar to NOAA. And their group, Emmanuel DeVred's group, they actually are they're trying to actually figure out what type of they have ways of figuring out what type of phytoplankton are there based on the satellite imagery. And so actually they do have some data on how the bloom changes from different types of organisms. So basically the issue is that I would need to get a C to N of everything that was there, an average C to N over everything that was there. And so obviously there's limits in these ranges, and I literally did that calculation just for you guys today because I like to connect it to carbon at some point. And I think in the papers we come out with, right now we've been kind of all like really plugging and chugging our own data, and this workshop is going to help the nitrogen people and the carbon people come together and be like, okay, what units are you using, and what is your, integrate over, and like what numbers can we do to compare. The other thing that I didn't really even mention in here is that we have a whole other host of work of a second model where we're looking at the ammonium isotopes and the particulate nitrogen isotopes, the N15PN. And we have another model that we're looking actually at the turnover ammonium that's happening while we're there. That, though, is a much shorter time scale. And so the models that we run for that are like, a couple days. Because the ammonium doesn't stick around, whereas the nitrate, it kind of integrates over the whole time. The ammonium doesn't. And that also might, be able to help us kind of answer some of the other types of questions that come from this bloom. But I think the nitrate isotopes I thought was just also a kind of cool way to like get some of these more integrated numbers. Yeah.
So, do you, it's very cool. Do you find a way to confirm your nitrification rates, for example, isotope dilution experiments, things like that?
Yeah.
And upper flux of nitrate, people have done that with helium.
Yes, so we did do rate incubations on board. So we did have those incubator farms and did that. And what we found basically is for the profiles, like it follows very similarly what we think. Now, what I haven't done is in terms of like, okay, we do know that there's tiny bits of nitrification happening at the very edge of the euphotic zone that probably could be supporting this, 6 that we're getting there. And in terms of the resupply, it's more that we've been looking at the diffusion and we've been looking at the, gosh, I'm like really blanking on the term, the gradient, like the nitrate gradient. So we're doing separate calculations of how much nitrate we expect to go just based on the density gradients. And that's honestly how we know what to test in our model. Does that make sense? So everything, like we always start at numbers and try and get to the isotope profiles, but some of the ways that we start at those numbers is based on things that we can already measure easily, if that makes sense. And so some of those numbers, like it isn't from the isotopes alone, right? Like we do have to do some of these other measurements to help us out. I always have this discussion, like could we get to a point where we don't need the incubations at all? Because they really do help us ground the models and kind of know where to start. It's like, I think so. It's just that model is just going to have a ton more uncertainty without having some of the incubations in there as like at least helping us place things in the water column and what we think like somewhat the rates should be around. Yeah. Okay. Oh, yeah, go.
You didn't mention anything about what concentrations of nitrate or any of the things you're actually using. And the reason I ask is because we've done similar experiments and we found that the fractionation factor is dependent on the concentration.
Yes.
And so I just wondered, like in these upwelling areas, obviously going to have much higher concentrations.
Yeah.
Than in other areas, and therefore the fractionation is different.
And that's the one thing that so I've had this thought of, because basically what I feel like you're saying is saying, okay, so so we can go back and look at the concentration in these purple, it was right there. So it gets up to about 15 at 100. And so our actually model only goes to 100 meters, and then it goes down to somewhere between 0 and 5, so like 2. So I feel like even what you're saying is what we've thought about is having a varying isotope effect with depth. Because if it is concentration dependent, then it should kind of vary with depth and potentially time.
So there, in fact, at like 1 micromole or something, there's no fractionation essentially. But at 5 micromoles or 10 or even higher, you get up to value.
Yeah,
And
yeah.
that's why if you look at the literature, there's so many different values.
Because it depends on what they did their incubation at. Yeah.
Amounts.
Yeah, so I think like we're thinking of our isotope effect as a, as like an apparent, like what you see, not really what's happening in each time. And so I think we're assuming that on a whole, it's happening near 5 per mil. Now maybe it's not. Also down here, I mean, most of the phytoplankton are doing the uptake up here. They're not doing it down here. The majority of because there's more light up here.
So the 5 per mil is actually exactly what we calculated for concentrations with high nitrate.
Nitrate, yeah.
And and they go to 1 essentially when nitrate is limiting.
Yeah.
Because they're just grabbing everything they can.
Yeah. So that's a really good point. So basically almost what you're saying is like in this part of the bloom, it'll be 5, but as you get to this part of the bloom, it might be lower. And so that and that even could be what this 4.4 number we get is the fact that like, yeah, for a certain amount of time when there's more nitrogen there it's 5, but like by the time most of the nitrogen's gone, it's yeah.
So then if you, like on that graph there,
Yeah.
in fact, the linear relationship which you're calculating alpha from should be actually a curvy linear because basically it changes.
Yeah, and
as you're
actually,
rolling it down.
there is a slight curve to this. It's kind of hard to see there, but the model actually does predict a slight curve. This line is obviously, we just put it in there. But yeah, I mean, it's a good point. I think there could be a way that the student could try and have this be a changing isotope effect with concentration. I don't actually think that would be that hard to build into the model. Yeah. Okay. So now I'm going to introduce my other project, which is it won't be as data heavy as that, but it is just an idea and work that we're doing. And if people have ideas. So this is work for my student, Zach. He's just started his PhD. Well, I guess he did 1 year. And this is working in Bedford Basin. So the data I showed you earlier was in this northwest arm. And now we're talking about data all throughout this harbor in Bedford Basin. Just to place this, here's Nova Scotia. I guess I should have had all of US so you could see where Nova Scotia is related to Florida. It's way more north. Halifax is right in the middle. Here's Dalhousie. And then obviously here's Bedford Basin. If we look at a cross section of Bedford Basin, what it looks like is it actually is a pretty deep basin of about 70 meters. And you kind of get this surface layer and deep layer. And so a lot of, like, the tides come in, but it takes a certain amount of wind and storms to create overturning of this deep layer. So this deep layer in time does become hypoxic, and I'll show some of that data. So this basin has been studied for years and years and years. There's a weekly monitoring program through the Department of Fisheries and Oceans that's been operating there for a long time. So we know a pretty good amount of what is happening in this basin nitrogen-wise. So this was a paper from 2021 by a student, Sebastian Hawes, in our department. And he looked at this weekly time series. And so you can see the seasonal cycle that happens. Temperature goes up in the summer, and then you see these dotted lines are basically when you get these, they call them oxygen intrusions, but basically when you get enough mixing that the deep water mix and oxygen's replenished. But before that, those mixing events, you can see you get these hypoxic conditions. So this is red, is just showing lower oxygen at the end of the summer, after you've had a lot of nitrate being taken up here, and then the organism sinking to the bottom, consuming the oxygen, and then you have these replenishment events that bring the oxygen back into that deep basin. He did a model looking at how the nitrogen cycle kind of changed along with these intrusions. So in blue, you can see ammonium, and yellow in nitrate, and then the red is nitrite. And so what you can see is that ammonium would actually kind of build up until you got ammonium oxidation to nitrite, and then you get a little production of nitrite, and then sometimes that would build up less, and you then get the nitrite oxidation to nitrate. And you'd have these kind of, we call it a nitrification cascade that would happen each year. until the whole system was mixed again, and then all of a sudden you got the buildup of ammonium. And so you can see in the blue, you can see how these mixing events were changing the oxygen. In his model, he actually showed how ammonium archaea and bacteria, how different organisms and then nitrite oxidizing bacteria grew in the basin as it was following these nutrients. So again, like, it's a really interesting system for nitrification. But he was looking at one spot of the basin and was really thinking about how the microbes were living there. The other thing is that they've been measuring N2O at this monitoring site for a long time. This is not the best plot, but I got it. I should have put the credit is from the Cirque Ocean Lab, or Doug Wallace's lab at Dalhousie. So they've been monitoring the N2O there for a long time. And so the black dotted line is the oxygen. The red and blue is the nitrous oxide at the surface and at depth. And you can see that, oxygen drops and you sometimes get these small little bits of N2O. But then sometimes if oxygen drops even further, you get a little bit more N2O. This is COVID. And now here you get the lowest drop of oxygen we had in 2021. And then you get this huge peak of N2O, up to 600 nanomoles of N2O. And so you can see there is this very tight relationship between oxygen and N2O in the basin. And there's still, we think this is could be due to nitrification, or we don't actually know yet, I guess, whether it's due to nitrification or denitrification. The oxygen never went to zero. So it could be from nitrification, because you do find that more N2O is produced as a side product when there's less oxygen. But we're still they're still working on figuring out. We have a lot of the eDNA, but I think that's why from the eDNA they were thinking it was an ammonium oxidizer. Using that. So we kind of know something about what is happening there, but I really want to ask the question is the nitrogen cycle there, how much is it being supported by the fact that you have all this anthropogenic nitrogen coming in? So we started a sampling program that's now looking more detailed at what nitrogen sources are to the basin. So my students started sampling weekly at Sackville River. So Sackville River is an estuary that has a lot of, could have a lot of fertilizer runoff, a couple other wastewater treatment plants further north, and other human activities that would be drained into the basin. We also have these 3 wastewater facilities. Dartmouth and Halifax have much, much higher amounts of sewage flow than Mill Cove. These are both what we call advanced primary treatment, which means the wastewater comes in, they add a coagulant, and so the solids stick out. And then for half of the year, from May to October, so the summer months, they UV that effluent, and then it goes back out. It only spends about 30 hours in the plant. And so basically all the nitrogen that came in comes out. And we've been measuring this. So like millimolar levels of ammonium come in and millimolar levels of ammonium go out. Mill Cove is actually the only plant that has actually some secondary treatment. And secondary treatment is where they have extra tanks where they add oxygen and they're trying to promote nitrification. And so you can see some of that ammonium getting converted to nitrate. That is not always the case. We've definitely seen pretty high levels of ammonium coming out of this plant, even though they have that secondary treatment. But again, no tertiary treatment. So all the nitrogen that came in from the waste is going straight into the basin. Lastly, and I think this is like what's kind of topical at the time, so we've been studying Bedford Basin, then a company, Planetary Technology, comes in, and they have now been adding alkalinity to the ocean. And so Ocean Alkalinity Addition is a way to sequester carbon. I'm going to go through how that works in the next slide. But basically, they're doing it at Tufts Cove, which is where our nuclear power plant is, and it's actually cooling. It's not a nuclear power plant, sorry, just power plant is where it's cooling the towers, and they use the water to add. So they have this tank of alkaline materials, and it's coming out in a slurry, and they're just basically using their pipes. And because they're using their pipes, they actually can get it. It's really easy permitting, because basically the permitting is very similar to the permitting that that the power plant has to do for releasing the warmed water into the environment. And so it's different than if you tried to do this, off of a boat. So basically, we want to use our isotopes to track the effects of these human activities on the nitrogen cycle. So I kind of want to know how much of that nitrification cascade, for example, that Sebastian was measuring, how much of that actually was due to the fact that there's loads and loads of ammonium coming into the basin that wouldn't be there naturally, and we're pretty sure it's actually a huge portion of the primary production and things going on in the basin is controlled by these things. So the research program now is kind of being built. We're getting these data sets right now. So we get weekly sampling at Sackville River. We get weekly sampling at the Bedford Basin Center during the time series. Now we have these monthly surveys. On the monthly surveys, we've been also sampling at the diffuser pipes in the basin and in a few other places. And these monthly surveys were actually set up to track what was going on in Tufts Cove. And then we've also started working with Halifax Water. It actually took me years and years and years to kind of it's one of those things, it's like a delicate relationship to build, where they now actually give me their effluent samples to look at the ammonium concentration, nitrate concentrations, and the isotopes. So now we're going to get a really good signature of what the wastewater coming in is. And we're getting these bi-weekly, so because this actually does change a lot based on rainstorms, because all the water is coming into these facilities, and so they really see kind of pretty fluctuating levels of sewage in there. So this is everything together. So the sample types that we're getting is all the nutrient concentrations, and then we're going to be getting the isotope signatures of the nitrate and ammonium, and then we're going to be getting N2O concentrations. And I already kind of showed you some of the data that we have. And so the idea is to use all of this together to build these models to say, okay, how much of the primary production and nitrification is being fueled by anthropogenic nitrogen, and then how is ocean alkalinity enhancement maybe changing some of the microbial processes that are already going on there. So I just want to end with why I think that there could be some effects to the nitrogen cycle for ocean alkalinity enhancement. And I think it's really important because I actually think it should be something that they should be considering when they're calculating their carbon credits. Because if they're producing things like N2O or making any effects that stop the amount of biological carbon uptake that's already going on there, then they shouldn't really be receiving as many credits for what they're doing. Okay, so basically the way that ocean alkalinity enhancement works is you add in an alkaline material, so you increase the alkalinity of the ocean. At the same time, you have local increase of pH. What that does is it draws in some CO2, and the CO2 is converted to bicarb, and it's stored as bicarb in the deep ocean. So the way that Planetary is doing it is through a pipe. There are other ways to do it, spreading it on beaches, off boats, doing electrochemical separation. They are calculating how much carbon is taken from the atmosphere due to adding this alkalinity, and that's how they get their carbon credits. They have to subtract any carbon they burned to get the alkalinity, the alkaline materials there. But they do not have to consider, they have to make sure, they have to show that they're not impacting the environment, but they're actually not using that. That's just so that you're not ruining fisheries or the lobster fisheries or the things people care about and killing things. But they're actually not really required. They use a term called MRV, which means monitoring, reporting, and verification. And that is built on models that are actually modeling the CO2 uptake and the advection of this extra bicarbonate into the deep ocean. But what I've noticed is that the biological impacts is thought of as a separate thing, and it's not part of the MRV. And I'm trying to say, okay, well, if you're changing how much carbon the ocean is taking up naturally, then and if you're producing more greenhouse gases, then it should be part of the MRV. So nitrification, so we've already kind of talked about nitrification. We think of it as ammonium to nitrite to nitrate. There's actually a lot of literature saying that they're actually doing the oxidation on the ammonia, and that they have ways for taking off a hydrogen ion, or changing their intercellular pH to make it ammonia, and what they're actually oxidizing is the ammonia. it's important because that's where we'll get at how this could be affected by ocean alkalinity enhancement. Either way, I also told you that they produce N2O, and this is something that could really change the climate impacts of what they're doing. And so I also think we should just call it marine greenhouse gas removal, just so that N2O is always brought. When you're doing things in the ocean and we know it's a place where N2O can be produced, they should just bring it into the conversation right away. And I know this is trying to happen for methane. People have also talked about N2O during the iron fertilization experiments. That was a big worry that happened there, is like, are they creating more N2O? So people know about it. I just know right now in the protocols that these companies have to follow, they do not need to monitor their own N2O or pay attention to it. So the two things that I think could change for nitrification is, one, these changes in pH, and actually in where we are, they're adding a slurry that does have particles in it, and they are adding turbidity. The metrics that they have for when they can dose and not dose is actually, they're monitoring very closely their pH and their turbidity. And if they go over certain TSS amounts or any over pH, they have to stop immediately. So those are the two things that they are monitoring, and we know what levels they're allowed to do. Okay, so will these affect nitrification and therefore N2O production? So this is just showing the speciation, it's a Bejeron plot, so it's showing a speciation of ammonium. And you can see the pK is at about 9.2. Oceanic pH, you're mostly at ammonium, very little ammonia. Their target is up to 9. And then so now you're in a situation that has a lot more ammonia and less ammonium. Because of this, there's still a lot of research on this. If that's something that actually favors these nitrifiers, it could actually help nitrification. And then secondly, because nitrifiers are inhibited by light, phytoplankton need light, they are both competing for this ammonium in this surface ocean. If you block out light, you could be favoring nitrifiers over the phytoplankton. The other thing I should mention, a lot of this is really important for places like Bedford Basin where we have very high levels of ammonium. So if you block light, you can favor the nitrifiers, make more N2O, and actually turn off some of the biological carbon uptake by phytoplankton. Okay, so that pretty much ends my talk. So really our ongoing future work, besides just looking at these data sets, so we're gonna have these very complete data sets to kind of look at some of these things. They've been dosing for 2 years now. We have, so we've had this monitoring for a bit, and we're gonna do a lot more of these kind of isotope models to get at what's happening with nitrification there. And also looking a lot at the N2O production. But we're also setting up laboratory incubation. So we have these mesocosms to specifically look at kind of the competition between phytoplankton and nitrifiers with adding turbidity and using the actual particles that they use, and also doing some culture incubations to try and understand a little bit more about how pH affects N2O production and the growth of these nitrifiers. So that just concludes my talk. I guess I always kind of end, this is kind of a little summary of the research in my lab. You can see we do a lot of diverse research. I talked about Bedford Basin and Labrador Sea. I kind of plugged kelp aquaculture and working with cultures. We also go on biannual cruises to the Scotian Shelf, and we have actually a lot of interesting data how nutrient dynamics are changing on Scotian Shelf because of climate change. And then also actually we have some work up in the high Arctic looking at nitrogen coming from glacial and impacted areas and fjords up there. And then, we build these are our tools. We build models and we fingerprint. So I guess, I have some acknowledgements. I want to acknowledge, so all my stuff is measured in the CERC laboratory, and Claire, she's the tech, and so she really makes a lot of this happen. And I think in chemical oceanography, all of us know that some of our most important people in our lab are marine technicians. All the students, I highlighted them throughout. And then my funding sources, which is NSERC, that's kind of our NSF, and then the Ocean Frontier Institute, and they're who got this big amount of funding that has been supporting all of this work in ocean alkalinity enhancement research. So, okay, any questions?
So I guess I have a two-part question.
Yeah.
First, how are you measuring the nitrogen and oxygen isotopes of nitrate? I assume, given that you worked with Karen, that it was the denitrifier method, but I would ask.
Yeah.
So for your second case study that you presented, have you thought at all about adding like a third dimension to your isotope plots, potentially the cap delta 17 measurements, or looking at a quote unquote co-contaminant that you could add to the chemical fingerprint?
That's a really good idea. So we use the denitrifier method exactly. For that work, I have not thought about adding a third, because actually, honestly, there's a lot. It was a master's project, and I would say it's just like at the very, or I guess you're thinking of for the Bedford Basin stuff. Yeah. Okay. So, the truth is that when we build these models, we already have like 6 parameters, but it's true. I feel like there could be a point where we would want to add another tracer, and our system actually doesn't measure cap delta 17. We don't have the capacity for doing that. The other thing that we can't do is for our N2O isotopes, we can't do site preference, but we actually are in the process. We just put in a big grant to try and get a new mass spectrometer, which would maybe be able to do some of these other things. But I would say, maybe like those would be the next steps because we're going to come out, these models are great. They're really fun to test things, and you can get really good ideas of what's going on, but you always come out with a lot of uncertainties where you're like, it could be this or could be that. And that's where maybe these other tracers would come in and be like, okay, we really need something else to help us separate these processes. Yeah. Yeah.
Great talk.
Thanks.
I'm really interested in the pH control on ammonium oxidation because it's something we need to think about with depth in a natural water column. So I think this is a really cool application of the same problem.
Yeah.
I'm trying to remember if I'm thinking about this correctly. Ammonium is transported actively, but ammonia should diffuse freely.
Exactly.
So actually, they might not benefit from the increase in ammonia because it's going to diffuse out just as much as it's going to diffuse in.
Oh yeah, that's true.
So I think you could do that in a model, right?
Yeah, that's a really good idea. Yeah, because that right, exactly. So they have only the transferter for the ammonium, but then if there's not a lot of ammonium, they have to convert it to ammonia and then oxidize it. But if there was a lot of ammonia, then they don't need to transport it and they just let it diffuse in and diffuse out. Yeah.
But it's just like we think about for carbon fixation, CO2 is leaving just as much as it's coming in. So,
yeah, that would be really easy to test in a model. And it probably would make sense to do along with some of these incubations just to see if it follows what we think it should. Yeah, that's a really good idea. Yeah.
Carly, is there temperature sensitivities for nitrifiers?
Oh, that's a really good question. We grow them at room temperature. My guess is that there probably is.
Polar waters, you see the highest ammonia concentrations, right?
Yeah.
Like if you look in the Ross Sea after a long winter, ammonium's still micromolar concentrations down there. I'm just wondering if nitrification contribution is changing through your season as a consequence of warming waters versus starting cold.
Yeah, I don't know. I mean, I'm sure I would have to go back at the original literature with a lot of these cultures. My guess is that they've grown them they do probably grow at a pretty wide range of temperatures. But I don't know at the thresholds if you would see it in a seasonal.
Real cold waters, you find high ammonia, even in deep waters where there's
So you think it could be, and that would mean it's maybe limit, that's something that's keeping them from taking it all the way down.
I think it shuts it down at really cold temperatures. And I'm guessing it's cold there.
Although the highest levels of ammonium that is in the winter. You're right. Yeah. So you're so it's we wait till it warms up and then they turn on and then go.
So your nitrification contribution would increase as you warm up.
Yeah.
And there'd be a threshold where it would take off, right?
I bet that is to do with the seasonal cycle somewhat. Yeah. That makes a lot of sense. Yeah, I can.
One question. So it was really neat to hear about the relationships you've been able to build with the wastewater treatment and I got to hear something about the kelp farming. How has it been, what was it like building, how is the working relationship with Planetarian with the Ocean Alkalinity Enhancement?
Oh, actually, that's really interesting. So there is a group at Dow that is very tightly aligned. They have a big amount of philanthropy funding through Carbon to Sea is the program. So maybe some of you guys have heard that they're working very closely with planetary technology. Okay, so they're a big part of doing this MRV modeling. Now, a bunch of us got this other funding, this Ocean Frontier Institute funding, which is kind of our own funding. And we've been adopted into the group. So like we have weekly meetings and we're on the cruises, but it is a different relationship because I'm actually pretty much completely separate from Planetary. So they're like kind of more closely, but when Planetary publishes stuff, like my name won't be in there because I'm not someone on their original project. So I've actually tried to get we have a really cool program called MyTax. Actually, if any of you guys are interested in working in this space between industry and science and are looking for postdocs, it's a really cool opportunity for postdocs. So I've actually contacted Plant, and so what it is the company pays for half the postdoc. The other, the funding source pays for the other half, and I pay nothing except for advise and do the research. And so I thought like Planetary might be interested in a postdoc, and kind of their comments to me when I bring up nitrogen stuff, they're just like, it's not of high priority right now. And basically it's not going to be of high priority to them until someone tells them they have to monitor it. So that's where I'm kind of like, and I think outside research funding is trying to be like, okay, let's figure out what are things we should be making them monitor, And I do think like there could be a scenario that like, as long as they're staying at the pH levels they're at and at the TSS levels they're at, like there is a scenario that it's totally fine. I'm just not okay assuming that without actually like figuring it out. But either way, I would say like right now, a lot of these CDR companies rely on scientists. So they really are relying on the modeling for the MRV, like these nearshore ROMS models. So that's with Katja Fennell's group at Dow, is that that's who they're working kind of close with for actually modeling these simulations. And it's kind of what I actually showed in your lab class the other day. So I would say like they do want the best science. It's just that they still are going to work on their priorities, but it's not hostile at all, I guess. And I think I feel like I've seen that. Like I've been to a lot of the conferences where there's companies and non-companies, and I do feel like it's a field where they kind of recognize they're relying on scientists to help them actually do this MRV. The wastewater people, it's like very different. They're kind of just like, they're kind of at overcapacity in terms of like, their people are very overwhelmed and they don't really have time to talk to researchers, and like we just want data from them. So they're kind of like, why, like you basically have to convince them why your data is going to help them kind of thing. And so they're at a point where actually these wastewater treatment plants are going to have to get refit. They're planning for it for 2040. So it's like a 15-year process, and probably by the time we get there, like all the technology is going to be out of date, but just how it works because everything's really slow. So basically you have to kind of sell why what you're going to measure is going to help them, or else like they're really just not going to spend the time to give you the data. But really for me, there's an engineering group at Dow that's working very close with them. They actually have a pilot plant within the plant that they're using to test how they want the new plants to work. And so it's kind of like slowly making that relationship, and then they have the relationship with Halifax Water, and then getting the things. But overall, like it's a space that I'm definitely getting used to working in, but it feels like maybe more common in Canada, and maybe in the US it'll turn more that way, where you really do have to have these applied partnerships. But yeah, the MyTax for students that are not sure is actually a really good way to like experiment whether you would like working with a company or not. Yeah.
Is there a pollution problem there with all that millimoles of ammonia and whatever they're checking out from those sewage plants? Or
the thing is like there was no wastewater treatment there until 2008. So people are very used to that bay being completely disgusting. So like raw sewage used to go in there all the time. And I think this is how also Planetary got away with dumping alkalinity in there, because no one really sees that as like, you're going to ruin our ocean. Like it's already surrounded by all this stuff. So yeah, like I would say, yes, the basin can look quite dirty. But do we have harmful algal blooms? Is it affecting, no one's actually fishing for mussels in there. So I don't think it's not extending any further than.
You don't go up the coast and you don't get sort of macroalgal bloom.
Not really. So that's the thing, is I think it's diluted once it gets out of the basin. It's diluted so fast that it's still, I mean, we're still a lot less people, right? And we're still like more ocean. So I think overall, it's just not the eutrophication issues that you'd have at like the base of the Mississippi or like, you know, around here if they dumped raw sewage into the ocean. But time will tell. So maybe in warming and in pollution, like the population of Halifax is growing hugely. So maybe eventually it would get there. And that's something I've like kind of thought about, like what would be the tipping point? Because the other thing is we have a ton of finfish aquaculture. We have a ton of salmon aquaculture all up and down the coast. And those two, you know, talking to the group talking to John over there, is like, so far the salmon, you know, they're dumping tons of nitrogen in the ocean, but it dilutes so fast. And so it really isn't causing these issues that you might expect other places to have. So it's still, we're lucky in that like, we're at a point where like, maybe we could stop it before it becomes a big problem, if it would. Yeah.
Any last questions you want to throw out?
Okay.
And again, Carly will be here, so let me know.
Yeah,
if
I'll be
you
here all
want to
next
meet with
week.
her.
So yeah, let me know.